Incubator flexibility plays a critical role in optimizing HEK293 cell cultures for gene therapy applications. And as gene therapy evolves, harnessing the full potential of HEK293 cells, known for their efficiency in transfection and viral vector support, becomes crucial. I'm Miles Skoda. This is the Life Science Connect webinar series, and today we're talking HEK293 cells for gene therapy, the role of incubator flexibility. Now, joining me for this presentation are two industry experts. First, from the Jefferson Institute for Bioprocessing, Associate Scientist Rui De Paula, and from INFORS HT, Regional Sales Manager Mike Lesh, who together are going to discuss the role of HEK293 cells in gene therapy and their application in viral vector production, cover how multi-cabinet incubators with adaptable parameters can optimize cell culture conditions, look at the impact of relative humidity on HEK293 cell growth and production efficiency, and explore the methods used to achieve optimal conditions for viral production, including temperature and orbital diameter adjustments. Now, we'll be holding a Q&A session following this presentation. Please feel free to submit questions as we go along. We'll get to as many as possible during the Q&A, and any that we don't get to, we'll follow up on after the event. And then just a bit of housekeeping: if you run into any issues today, please try refreshing your browser first. If your problem persists, submit a question through that Q&A, and I'll help get you back on track. And then finally, this event will be available on demand, usually within a day, often less, and you'll get an email with a link to it, or you can just use the same link that you used today. So with that said, I'm going to turn it over to Rui to get started.
All right. Thanks a lot for the introduction, Miles. Really good to be here today for this discussion. So these are the topics we're going to discuss today. We're talking about the use of viral vectors for gene therapy, and what are the bottlenecks to make this technology viable and scalable. And then we're going to show some results that we got in this partnership between JIB and INFORS HT for a suspension HEK293 cell culture using the Multitron INFORS incubator. And finally, in the end, we have some time for questions and discussions, as Miles said. So let's begin. Talking about viral vectors for gene therapy now. One way to perform gene therapy is to use viral vectors such as adenoviruses or lentiviruses. So these are viruses that, when they infect a target cell, they can insert specific genes that will have a therapeutic effect on the patient. And this therapy can be done in vivo. That's when the viral particles are injected directly into the patient, or ex vivo. That's when the cells are removed from the patient, transformed in the lab, and then re-injected into the patient. And of course, each method has its own advantages and disadvantages. But what they have in common is that we need to have a production of viral particles that's technically and economically viable, right? In this table, we can check the number of clinical trials reported for each type of viral vector for gene therapy. And we can see that adenoviruses, they are the most relevant in this case, with 575 clinical trials happening back in 2021. That's the date for this reference. All right, so the production of viral particles for gene therapy requires a host cell, right? A cell that, when we do what we call transfection, produces viral particles with high efficiency. And a cell used a lot in this case, it will be HEK293 cells, human embryonary kidney cells. And we have a lot of reasons to use HEK293 cells. We can say that they are well characterized for this application, and there is a lot of knowledge in the literature and among scientists and companies about how to do the transfection. And maybe even more important, the fact that we know that we need to use a human cell if we're going to produce viruses capable of infecting human cells when we go for the therapy itself. So probably companies and scientists, they would just love to use CHO cells for viral vector production, but then you would have a lot of problems because that's not a human cell line, right? But anyway, we can say that we still have some challenges in using HEK293 cells for this application, especially when we think about scaling up and really making this technology viable for companies and also for patients. Yeah. All right. So HEK293 cells, they are originally adherent. They prefer to grow on a surface where they are attached. For this, we use flasks like this or cell stacks like these ones in the lab if we want to work on a larger scale. And this usually works very well, especially when we think about the yield. So for example, what we call YPX, that's the yield, how much of your product, in this case, viral particles, each cell is producing. Okay, so that's the YPX. And usually that value would be higher when you work with adherent culture. But then on the other hand, adherent systems, they are difficult to scale up. And scaling up, that's really essential if we want to make gene therapy something accessible, meaning lower costs for the company and lower prices for the patient. So basically, scale up of adherent cultures happens in 2 dimensions, right? With area. You got to work with area, 2 dimensions. And then if you go for bioreactors, suspension bioreactors, like this one here, we're going to work with volume. So in other words, we'll have to use more clean room space, which is very expensive if you go for adherent systems. The process, it will require more labor, more scientists, or more operators working on the process. We have less control, we have less automation. You have all these problems to deal with if you go for adherent systems. And by the way, people with proper training to work with adherent cells, it's also not easy to find. It takes a lot of training and experience in aseptic techniques, right? So you have a lot of reasons to just go for a suspension culture if you can, right? So for example, in this reference here, we can get an idea of the difference between working with adherent culture and suspension culture. In this case, 600 cell stacks units, they produce similar amount of cells as a 200-liter suspension bioreactor. And then think about how many people you need working if you have to do a production using 600 cell stacks. And think about how many people you need to operate a 200 liters bioreactor. It's a big difference, right? So here in JIB, for example, we have a lot of experience in scaling up protein and monoclonal antibody production, using CHO cells in suspension. But so far, our experience with viral vectors has been mostly using HEK293 adherent cells. And that's something we want to change. We want to have a scalable viral vector production platform using HEK293 cells. So in other words, using HEK293 cells adapted for suspension. But that also means we have some additional variables to test, such as agitation, aeration, the orbit diameter, the shear stress. And these variables, they simply don't exist when you're talking about adherent systems, right? But then when you go for suspension culture, you have to deal with the more variables to optimize. So that's why this partnership with INFORS came at the perfect time for us here in JIB. If you're going to optimize a small-scale culture using shake flasks and need to test different variables at different levels, if you have an incubator where you can have different cabinets, right? So you have 3 cabinets on the Multitron INFORS incubator, and you can really have different set points on each cabinet, and that's really useful if you want to optimize a cell culture in suspension on small scale, like shake flasks. So, as I said, you can have different set points for temperature, humidity, agitation, CO2, and you can have different orbit diameter configurations for each cabinet, and it really makes your life just much easier in the lab. So yeah, here in JIB right now, the first step to really make a scalable, to really have a scalable viral vector production platform using HEK293 suspension cell lines, it's to test the cell culture, right, with different levels of these variables. In particular, we need to know which condition will provide us the highest cell density and also the highest specific growth rate. Because the timing and efficiency of the transfection process in a HEK293 cell culture for viral vector production really depends on these variables. Because usually the transfection, to have a high efficiency, you got to have a lot of cells, and you also need those cells to be really active. And that means you have to know which condition you have the highest specific growth rate. And then something else, we also investigated different positions of the flasks inside the shaker, right? So in the corners and in the center, to see if there would be any differences on the cell culture results. Yeah, so about the materials we used, we used that media from Gibco. It's a freestyle 293 expression medium with 2.5% FBS, 100 micrograms per ml heparin to avoid clumps, Glutamax, and also antibiotic, 1% volume penicillin streptomycin. We use that cell line. That's also a Gibco cell line 293F. It's a cell line adapted to suspension culture already. You have a superior transfection efficiency, and you can also have high protein expression levels. And of course, the equipment, the incubator we used, that was the Multitron incubator shaker. It's a triple stack system. You can have adjustable drive option, and you have humidity and CO2 control for each cabinet if you want at a different set point for each cabinet. So yeah, as I said before, the first step really, if you want to have a suspension HEK293 cell culture platform for viral vector production, is to find the best condition to do the cell culture with your cell line. And each cell line will have a different oxygen demand. Each cell line will have a different sensitivity to shear stress. So here we can check the experimental planning that we did so far here in JIB in this partnership with INFORS. Each line here will be the condition that you have on each cabinet for the incubator. The CO2 and the temperature, we just had the same values for all experiments, CO2 at 8% and temperature at 37 degrees Celsius. But we tested different levels of agitation, orbit diameter, and humidity. We can say that higher agitation usually increases the mixing and the aeration inside the shake flask, but it also increases the shear stress. So what about the orbit diameter? We also call this the flow. And that means what will be the diameter in the orbit movement that the flask will be doing inside the shaker, inside the incubator. And a higher value for the orbit diameter means more mixing and more aeration, but it also means more shear stress. And finally, the humidity. Sometimes we don't even worry about the humidity level that you have in the incubator, as long as you have some to avoid evaporation. But in fact, that might be an important parameter, because a humid environment with controlled humidity inside the incubator can help promote the diffusion of gases, meaning the aeration that happens through the surface in a shake flask culture. So that might be an important parameter to have control inside the shaker. And yeah, for the experiments, all of them we used 1 liter plain vented shake flasks with 300 mLs working volume. Yeah, and then something else, we tested different positions for the shake flasks inside the incubator. One shake flask on each corner and also a flask at the center. Basically, inside each cabinet, you have the same conditions anywhere, but that's something that we wanted to verify to make sure, right? So having the same result, we can consider that all those flasks inside the cabinet, they were replicates. Yeah, and then for these experiments, we analyzed cell density and viability using the nucleo counter NC202. We analyzed glucose and lactate using the biochemical analyzer WISE, and osmolality using the Osmotec XT equipment. The osmolality measurement can help you understand if you have different levels of evaporation happening in your cell culture. And that makes sense since relative humidity was something that we tested in these experiments. All right, yeah, and that's just a picture taken during the experiment. For the bottom cabinet, we can clearly check the position for each shake flasks on the corners and in the center. All right, so finally, the results, okay? So here we can check the results for cell growth, viable cell density with time. For all these experiments, the different positions inside the cabinet, they really had similar results. So we just consider the flasks inside the same cabinet as replicates. And that's where we get the standard deviation in the graph. And we can see that we had some differences for each condition tested. The best one was really this one here, the blue line that used 110 RPM agitation, 25 millimeters orbit diameter, and 85% relative humidity. And if you compare with the other conditions, these are really the highest levels tested for each variable: agitation, orbit diameter, and relative humidity. Yeah, so now on that table, we can check the result for specific cell growth, calculated with the exponential phase of growth data for each condition, the doubling time, and also the viability that was analyzed on the last day of culture with 6 days. Yeah, so as I said before, when you are planning a viral vector production, you have to decide the transfection timing based on your cell culture results. Normally, we don't have a stable producer cell line for viral vector production, like usually happens with monoclonal antibodies production with CHO cells, for example. So to have our product, in this case, viral particles, we have to infect those HEK293 cells in culture. And then you have the question when to do it. It might change depending on the virus you're using, but usually you need a high cell density, and you also want your cells to be really active and doing cell division at a high rate. And that's why you want to check the specific growth rate to decide the best condition to use in this application. And in these experiments so far, the conditions with higher agitation, higher relative humidity, we really had the best results when you check the specific growth rate. And then about the viability, the results were mostly good and the same for all experiments. They all finished with viability levels close to 97, 98%. So that's a good thing. All right, so what can we say as conclusions? Within the tested limits, high values of agitation, 110 RPM, flow 25 millimeters, and humidity 85% relative humidity were beneficial for HEK293 cell culture in suspension with the cell line we're using, of course. We can say that within the same cabinet, no significant differences were found in the cultivation results for different positions of the flasks. The optimization of HEK293 cell culture to produce viral vectors presents additional complexities and variables to consider, demanding flexibility and reliability in the incubator to be used. And you really have a big advantage in using a multi-cabinet incubator where each cabinet can have a different set point for temperature, CO2, agitation, and relative humidity. All right, so I just want to say a few words about JIB now. So JIB goes for Jefferson Institute for Bioprocessing. We are part of Thomas Jefferson University. Physically, we are like 40 minutes away from Center City, Philly, but we have a state-of-the-art facility for bioprocesses, teaching, training, and process development. We have several clean room labs with upstream and downstream equipment, both small scale and pilot scale, that we use for teaching, for training, and also we do research and development with partners. Yeah, and that's about the structure, okay? But we also have a lot of expertise in different areas. Cell culture, monoclonal antibody production, upstream, downstream, cell therapy with stem cells, T cells, gene therapy with viral vectors. So if you want to know more about us, please feel free to contact Aldo or Jeff. Okay, so Aldo for services, Jeff for academic, and that's it, right? So now, my friend Michael can speak a little more about INFORS HT, right?
Thank you very much, Rui, for your thorough and educational discussion. It has been great to work alongside you and your colleagues at JIB throughout this collaboration, and we're excited to work with you more in the future to better support our mutual clients. Now I'm going to take a few minutes to talk to you about INFORS HT, so you all have a better understanding of who we are and what we do to support the scientific community. INFORS HT was founded in 1965 in Basel, Switzerland by Alex Horolenko, who is pictured here, and today is still the owner of our privately run company. Since our humble beginnings, our organization has remained dedicated to developing innovative incubator shakers, bioreactors, and software to support the bioprocess workflow. We continue to grow around the world and have a strong presence supporting our brand and customers in the United States. Outside the US, we have many subsidiaries in Europe, South America, and 53 distributors worldwide. In the US, we have operated as INFORS USA since 2014. And we have a manufacturing and customer support facility just south of Baltimore, Maryland. Just like our founders, we work to help our customers simplify their workflows and make things better. We understand that better solutions will bring more cost-effective therapeutics to the market faster. So we take pride in helping our customers solve complex problems on a daily basis. All right, so here's a quick snapshot of the INFORS HT product portfolio. As mentioned, we develop and manufacture incubator shakers, bioreactors, and software to support the bioprocess workflow. We have solutions for microbial fermentation and mammalian and insect cell cultures grown in suspension. And looking at the diagram here, you can see that we have solutions from screening through production. From left to right, we have the stackable incubator shakers, and then we get into our bioreactors that offer tighter parameter controls. We can grow with you as you scale from the bench all the way up to 1,000-liter stainless steel steam-in-place systems. Our Eve bioprocess software allows you to unify process control and data management into a single bioprocess platform and can easily integrate third-party bioprocess equipment and data for more comprehensive batch planning, control, and reporting. It is our solution to further automate and digitalize your bioprocess. The Multitron is the incubator shaker that Rui and his colleagues at JIB are using for their HEK suspension cell cultures. The Multitron is an attractive solution for small-scale cultures using shake flask, as it provides flexible configuration options to optimize your cell culture conditions. With uniform temperature, agitation, humidity, and CO2 controls, you can expect consistent and reproducible results across the cabinet, regardless of flask or vessel placement. This picture shows the versatility of the multi-cabinet design. In the top unit, they are running flasks for protein production, but just below, they are using smaller vessels like plates, culture tubes, and bioreactor tubes for screening purposes. When working with many different vessel sizes, it is helpful to have the option to adjust and control the orbital diameter. As Rui previously mentioned, the orbital diameter of the flask from the center of the platform can impact aeration, mixing properties, and stress on the cells within the vessel, which can negatively or positively affect cell densities and growth rates. For these reasons, the orbital throw is an important parameter control to keep in mind when looking to optimize your HEK293 suspension cell cultures. Rui, I have a question for you. What has been your group's favorite design feature since you transitioned to the Multitron?
Well, I mean, these are all really good points, but I would say I like the ergonomics to get in and out of the shaker, the easy slide out tray, that's really helpful. Not only me, like everybody here in JIB really enjoyed that feature. Just makes our life easier when sampling and just handling things inside the incubator. Yeah, and I mean, something else, I really like how the humidity control works in the incubator. To have a separate vessel to feed water into the shaker, you don't have a tray of water inside the incubator like usually we see. And in my opinion, that's really just better when you think about how much easier it is to change the water. I also think it's a little cleaner inside the incubator. So yeah, I would say these features are the ones I really appreciate.
Thanks again, Rui. I'm happy to hear that your group likes the design and functionality of our humidity control system. Using sterile water to actively humidify the culture environment is very important to limit the risk of contamination. I'm also glad to hear that the ergonomics of getting in and out of the shakers are features that you and your colleagues appreciate. These visuals here show an ideal working height, even when stacking up to 3 high. The slide-out tray with automatic lock and unlock mechanisms makes all flasks easily accessible when you need to take a quick look at the cells, do a phase change, or make a media addition. We are getting close to the end of the discussion before we leave some time for questions and answers, but just a few more things worth mentioning about the Multitron. The interior design mitigates contamination risks and makes routine wipe downs, even under the table, easy. This picture shows the rounded corners of the interior and provides a look underneath the table, which is also easily accessible to clean and disinfect as needed. We want to keep downtime to a minimum, so we always have that in mind with our design features. Lastly, the touchscreen display is intuitive and simple to use, so parameter changes can be done quickly. One of the new available features that we have added to the touchscreen display, at the request of our customers, is the event list. With the event list, you can quickly access the last 18 events that have occurred on the machine. Just a quick look will show you parameter changes, alarms, and door openings, which is super helpful to understand what's happening inside the incubator. And Rui, I have one more question for you. Can you name a time that the event list has been helpful for your group to use?
Yeah. Well, you know, Mike, everybody that works with cell culture knows that eventually your experiment will go through the weekend, holidays. If you have a fed batch experiment, for example, it might be something like 12 to 15 days total. So it's really useful to have that touch screen with the history, so you can check any issues that might have happened and understand what happened, right? So let's say, for example, power outages, when the door was open, any deviations on the process values. Yeah, that's just a good feature.
Thank you again, Rui. I am glad that the event list provides your group some peace of mind and helps to keep your group connected when working with the incubators. To finish things off, I just want to make sure that you're all aware that we have local, dedicated, and experienced service teams across the globe to support your efforts and help to minimize downtime in the lab. We know our shakers are one of the many workhorses in the lab, and they're constantly running, so we take pride in providing quick and thorough response times to keep them running optimally. Whether it be routine preventative maintenance, repair work, service agreements, or calibration and testing services, our team can fully support you to make sure our equipment is running to manufacturer specifications. We are firm believers that once our equipment is in the lab, our relationship with our clients has just begun. At INFORS HT, we are here to support you from discovery through production, and we are very appreciative to work with collaborators like the Jefferson Institute for Bioprocess to help move science forward. Thank you all very much for your time today. Please let us know if you have any questions. We are happy to address them now.
All right, and with that, Mike, Rui, this was some great stuff. What an awesome presentation, and a lot of great data presented, and some really, really interesting things so far. We've got a few questions from the audience coming in. Audience members, just a few reminders. Right on your screen on the left side, kind of below where you're seeing me, there should be a place to submit your questions. Submit your questions now. We're going to get to as many as we can here in the little bit of time we have left. You should see that box being highlighted for you right now. Below that, just a note, there is our speakers information. So if you wanted to reach out to either of our speakers directly, you can connect with them on LinkedIn, or you can email them directly with the email address links, the little blue circle with the envelope inside. To the right of that, just a couple downloads for today, just about today's topic, some things relevant to what we spoke about, and feel free to download those. And then one last thing, I did have a survey that I set up on the right side of the screen. I'd really appreciate your responses too. I know several people filled it out already. Again, I appreciate it. Just some quick responses to the answers there. And I think you can just answer whatever is relevant. Don't have to answer everything in there. So let's see, let's get into these questions. Rui, I'm going to start with you on this one. So you mentioned that JIB is working on having a suspension culture platform for the production of viral vectors for gene therapy. Could you talk about what the next steps then are based on the results you showed today?
Yeah, thank you, Miles. That's a good question. So yeah, here in JIB, now that we know the conditions we need to use to have a high cell density with a high growth rate, right? For this cell line we're using, we have to test some things related to the infection efficiency to maximize the viral vector yield now. So as I said, we have a good method that works well with Adherent HEK293 cells using ADV, adenoviral vectors. But things, they might change a lot if you go for suspension, okay? Usually for better. So, for example, the mixing in a suspension system can make a more efficient distribution of the ADV particles, and that is going to promote greater contact between the ADV particles with cells, and that is going to increase the infections, okay? So that's what you need with ADV. If you have a platform with transfection instead of infection, the same thing will happen really, because your transfection reagents, they will mix better in the culture medium, and you probably have a increased cell reagent contact, and that is also going to increase the transfection. So yeah, you just have some more variables to test when you go for the infection or the transfection. And usually if you can save on these transfection reagents or the viral particles to use, it's really just great, because these guys, they are really expensive. And usually they are a bottleneck when you think about the costs on how to make viral vector production for gene therapy really viable.
All right. Yeah, perfect. Thanks, Rui. Let's see, up next. So Mike, I think, and this one's for you. And I think they're kind of talking about the Multitron here. So they were asking, does it connect to external monitoring devices or allow for remote monitoring and control? And then if so, kind of what are the options?
Yeah, that's a good question. So we do have interface options for both analog and digital. For analog connections, we can add an analog output box on the back of each unit. And with that, you can send 4 by 20 milliamp signal connections to your device. So you can send parameter values and set points to the device. And then for digital interface options, you can connect via Modbus or Profibus. And with that, you can actually monitor and control, because with those options, there are read-write capabilities.
All right, beautiful. Thanks, Mike. Let's get into a few more here. So do you think we can make small-scale lentivirus in 96 well plate with suspension HEK293 cells in the Multitron?
Yeah, I can take that question. So yes, you can. We do currently have a lot of customers that are doing that. We didn't speak to it too much in this presentation, but with the Multitron, we do have a 3 millimeter orbital throw option, and with that, you can agitate up to 1,000 RPMs. We have some studies that show that effective mixing in plates really starts around 800, 850 RPM. But with the 3 millimeter Multitron, we do have dedicated plate trays and other accessories for that application.
All right. Yeah, perfect. Thanks, Mike. Another one here, Mike, probably for you. So can you clean and lower the BioBird inside the incubator by using UV?
Yes, you can. So we do have a UV sterilization option with the Multitron. And how that works is UV light gets placed in the bottom of the cabinet, and it's going to sterilize external air that's coming into the cabinet. So it's going to clean the airflow coming into the cabinet. So that is the UV sterilization option on the Multitron.
Beautiful. Yeah, thanks, Mike. So in terms of, let's see, so could you talk about what kind of water you're using for the humidity? Is it distilled, tap, autoclaved?
Yeah, this is a really good question. A lot of times this gets overlooked. A lot of times customers will assume that they want to use the purest available water possible. So in most labs, deionized water or ultra pure water, a lot of people will call it milli-Q water in the lab. That's what customers will go for because it's the purest form of water available. But really that water is too pure. So we ask that customers use distilled, definitely not tapped. So distilled or reverse osmosis water is also acceptable. But those really pure forms of water, they have little to no ionic content in them. So they can actually kind of strip and break down the metal components of the humidity control system. And that can introduce contamination not only into, premature corrosion of those metal components, which then can introduce contamination into the culture environment. So we actually recently started making a optimized version of distilled water. And what we've done to that, we've adjusted the pH to 7 to 9. We've removed certain divalent ions in the water. And that water is optimized for a humidified culture environment. So that water is going to help to fight off any waterborne bacteria or fungi that might be formed in the water.
Perfect. Thanks, Mike. I think with that, we're actually going to wrap up here for the day. And so audience members, if you did have any more questions, you can definitely feel free to submit those. I will pass those along to the team here for potential follow-up after the event. And then again, you can reach out to our speakers directly with the bio information in the bottom left corner. But besides that, Mike, Rui, thanks so much for joining me today. Again, a great presentation, and thanks for getting all these great questions and getting through those. And audience members, we appreciate you being here today, appreciate your time, your attendance, and we hope that you all have a great rest of your day, and we'll see everybody next time. Bye all.
Thanks a lot, Miles.
Thanks. See ya. Have a good one.